In this video, I am going to explain about biomolecules. First, we will discuss synopsis points. So, this is lesson structure. So, here we will discuss carbohydrates, amino acids and proteins, enzymes, vitamins, nucleic acids and hormones. First, carbohydrates. The saccharides are sugars which can reduce failing solution and tolerance reagent are known as reducing sugars. So very important, reducing sugars. So in carbohydrates, these points are very important. Next, non-reducing sugars. The saccharides which cannot reduce failing solution and tolerance reagent are non-reducing. So remember, all mono and disaccharides except sucrose or reducing sugars. Very important. All monosaccharides and disaccharides except sucrose are always reducing. Next point. Invert sugars. So during hydrolysis of sucrose, the sign of specific rotation changes from D to L. This will be known as inversion. And the sucrose is also known as invert sugar. Inversion of cane sugar. Change in specific rotation from positive to negative. Next, lactose is also known as milk sugar. In milk it is present. Next, in cellulose, beta D2-glucose units are joined by beta 1,4-glycosidic linkage. Next, starch is mixture of amylose and amylopectin. These are the two components of starch. So, starch is polymer of, you remember these two. Starch, polymer of alpha glucose, and the cellulose is polymer of beta glucose. So these two are polysaccharides. Remember this. Next, anomers, a pair of stereoisomers which differ arrangement of hydrogen and OH at C1 carbon only of aldoses or C2 carbon of ketones are known as anomers. So very important. Here C1, C2 carbons are anomeric carbon atoms. So example for glucose, we have these two forms, alpha D glucose, beta D glucose. So glucose will be having pyranose ring structure. Now pyranose, 6-member ring. Next, glycosidic linkage. The linkage present between two monosaccharide units is glycosidic linkage. Next, maltose on hydrolysis will give 2 moles of alpha D glucose. Starch on hydrolysis by the action of diastase gives maltose. These are hydrolysis products. Next, glycosidic linkage present in amylose, C1C4 glucose, that is alpha type. Next, glycosidic linkage between two units of amylopectin are alpha C1C4, alpha C1C6. Next, amylose and amylopectin are parts of starch. Starch is polymer of alpha D glucose. Next, glycosidic linkage between monomers of cellulose, beta C1C4 glucoses, beta form. Next, So fructose is also known as levulose or fruit sugar. Next, mutarotation. The spontaneous change in specific rotation of optically active compound. So change in optical rotation. Spontaneous change must be there. Next, the functional groups present in typical carbohydrate are CO and OH. So CO is carbonyl group, and OH is hydroxy, that is alcoholic group. These are the functional groups. So here carbonyl group may be aldehyde or ketone. Next, amino acids and proteins, main points. So amino acids are the compounds which contain both NH2 and COOH groups. Next, proteins. Proteins are the polymers of alpha amino acids. Alpha amino acids. So proteins examples: hair, silk, insulin, albumin, egg albumin. Zwitter ion, the ion which carries positive charge at one end and a negative charge on other end, is Zwitter ion. So two types. That means let's structure like this. If you take glycine, it will be converted to, so here minus and plus. COH is acid. This reacts with lone pair of nitrogen to form Zwitter ion. Minus CH2 NH3 plus. This is Zwitter ion or dipolar ion. Zwitter ion or dipolar ion or inner salt. Inner salt. Remember this. Next, essential and non-essential amino acids. This list is very, very important. The amino acids which cannot be synthesized by body are essential. Non-essential, the amino acids which can be synthesized by body. Here examples are very, very important. Next, peptide linkase. CONH linkase will be known as peptide linkase. CONH. Peptide linkase. CONH. Next, globular proteins. The proteins in which chains of polypeptide coil around to give spherical shape. Globular means spherical shape. Fibrous proteins. The proteins in which polypeptide chains run in a parallel directions, held together with hydrogen bonds, are fibrous proteins. So in hydrogen bonds and disulfide bonds will be present. Next, denaturation. Denaturation means breaking of highly organized protein, ordered protein, and simultaneous loss of biological activity of protein. So denaturation means simply loss of activity of protein. Next. So proteins are nothing but polypeptides. Proteins are also known as polypeptides. Next. Enzymes. So enzymes are biocatalysts which are produced by living cells. Next. So enzymes which catalyze the oxidation of one substrate with simultaneous reduction of other substrate are named as oxidoreductase enzymes. So here both the reactions are happening, oxidation and reduction. So maltase hydrolyzes maltose to glucose. So maltase is enzyme. Maltose is disaccharide. Glucose is monosaccharide. Hydrolysis of maltose into glucose can be carried out by maltase enzyme. Next, vitamins. So vitamins are naturally occurring carbon compounds with low molecular weight. So these are essential dietary factors. Next, see here, these are required in minute quantities for the maintenance of normal health of organisms. So minute quantities are sufficient. Next, vitamin A deficiency causes night blindness. Vitamin A is also known as retinol. Vitamin D is known as calciferol. Remember here names. In matchings, these will be important. Next, vitamin E is also known as tocopherol. Vitamin E deficiency causes sterility. Vitamin K is anti-hemorrhage. Vitamin K deficiency causes blood coagulation prevention of blood coagulation. Next, vitamin D deficiency causes rickets. Vitamin B12, cyanocobalamin. Cobalt is present here. Next, vitamin C. Deficiency of vitamin C causes scurvy, bleeding of gums. Fat-soluble vitamins are A, D, E, K, and water-soluble are B and C. Vitamin B1 is thiamine, B2 is riboflavin. Next, B6 is pyridoxine. Chemical name of vitamin C is ascorbic acid. Deficiency of vitamin B12 causes pernicious anemia. Next, nucleic acids. Very important. Nucleoside is composed of base and sugar. So a sugar is pentose sugar and base is nitrogenous base, nitrogen containing base. Nucleotide is composed of base plus sugar plus phosphate ion. 3 components. Next, nitrogenous base not present in RNA is thymine. RNA means ribonucleic acid, we know. Next, so phosphate ester of nucleoside is known as nucleotide. Next, replication or duplication. Duplication of single DNA molecule into 2 daughter DNA molecules. Next, hydrogen bonding is responsible for double helical structure of DNA. So DNA shape will be double helical structure that will be stabilized by hydrogen bonds. Next, guanine, cytosine are present in both DNA and RNA. Next, RNA different from that of DNA because RNA contains ribose sugar and uracil. So, pyrimidine bases present in DNA are cytosine, thymine. Pyrimidine base present in both DNA, RNA, cytosine. Hormones. Hormones are the biomolecules of plant and animal origin that transfer biological information from one group of cells to distant tissues or organs. So, biological transformers are hormones. So, animal hormones are produced by specialized tissues called endocrine or ductless glands. Next, hormones are classified into 3 types: steroid hormones, polypeptide, amino acid derivatives. Examples are very important. Polypeptide hormones are very important. Insulin, oxytocin. Next, insulin decreases blood glucose levels. That means regulates. Hypothyroidism is characterized by letharginous and obesity. Letharginous and obesity. So these are the points. Next, last. Glucocorticoids controls the carbohydrate metabolism and regulates inflammatory reactions. Glucocorticoids. Next, insulin is best example for protein hormone. Protein. Next, progesterone helps implantation of fertilized egg. Next, estradiol controls menstrual cycle. So these are the main points of biomolecules. Next, we will discuss worksheet questions. See the first worksheet question. So very important. Which of the pairs represent D and L fructose? So D and L will be decided by C5 carbon. C5 carbon decides D and L configuration. This is last but one carbon or second carbon from last. Second carbon from last means from bottom. So DL configuration will be decided by this carbon only. So if this configuration is present, CH2OH, COH and H is present, this will be D configuration, D form. If this configuration is present, this is C5 carbon and this is C6 carbon. If it is like this, there is also C5 carbon, C6 carbon, L form. Check here now. So option 3 is D form, 4 is L form. That means 3 and 4. So OH right side and OH left side. So both glucose and fructose have same D configuration, which is correct. And both glucose and fructose are dextrorotatory. So glucose is dextro form, fructose levo form. So therefore A is true, but R is false. Option C. So dextro form means plus form, levo form minus form. Next. So denaturation of proteins can destroy all primary, secondary, tertiary protein structure. That is wrong. So denaturation destroys only secondary and tertiary structure. That means assertion wrong. Curding of milk is due to denaturation of proteins. R is true. Therefore, option D. Denaturation destroys only secondary and tertiary structure. Next. So, which of the following statements are correct? So, a carboxyhemoglobin must be there. Carboxyhemoglobin. Carboxy. Carboxyhemoglobin reaches to 3 to 4% in blood. So what is the effect? leads to headache and results in cardiovascular problems only. These two. That means option A clearly. Option A. Only headache and cardiovascular problems. Increase of carbon monoxide in blood up to 3 to 4%. Next. Identify nucleoside. Nucleoside. Side means sugar plus base. Nucleotide, sugar plus base plus phosphate. Remember the difference. Option A contains sugar and base. Next. So zwitterion from the following is zwitterion. Here clearly option D is zwitterion, which is CHCOO minus and NH3 plus. Zwitterion or dipolar ion or inner salt. These are other names. So both cation and anion are present in the same molecule. Therefore option D simple. Next. So which are the following are reducing sugars? So we know all mono and disaccharides except sucrose are reducing sugars. Very important. All mono and disaccharides except sucrose, which is disaccharide, are reducing sugars. See here, check option. Ribose, monosaccharide. 3. Lactose, disaccharide. 4. Fructose, monosaccharide. All are reducing. B option. Next. Insulin and glucosan, which are responsible to maintain blood glucose level, fall under. These are simply hormones. Insulin and glucosan are hormones. So biological messengers are hormones. Simply. Simple question. Next. So which are the following are reducing sugars? Now only we have discussed. All mono and disaccharides except sucrose are reducing sugars. So very important. So here except sucrose, maltose reducing, lactose reducing, fructose reducing. B, C, D. Option D. Sucrose is non-reducing. Next. So number of complementary hydrogen bonds between guanine and cytosine. So here ATGC ratio will be there. So between A and T, 2, G and C, 3. 2 hydrogen bonds. Here 3 hydrogen bonds. So between AT 2 hydrogen bonds, GC 3 hydrogen bonds. Option D. Direct question. So number of lines represent hydrogen bonds. Next. So thiamine is thiamine is thiamine vitamin B1. Vitamin B1 is thiamine. Simply option D. Next. In which of the following keratin, which is a fibrous protein, is absent? So keratin is part of hair, skin and nails. So in muscles it will not be present. Silk also contains keratin. Wool also contains keratin. Keratin is part of hair, skin and nails. In muscles it will be absent. Next. Curding of milk is caused due to formation of which of the following acids by bacteria? Here lactic acid. Lactic acid formula, remember, it will be formed by lactobacillus bacteria. Therefore, simply lactic acid. It is formed by lactobacillus bacteria. Next. So, pernicious anemia is caused due to deficiency of which vitamin? B12. B12 causes pernicious anemia. Simply option A. So, it is water soluble. Water soluble vitamin. And it is found in meat, fish, egg and curd, etc. Next. So which hormone tends to increase blood glucose level in humans? So here simply glucagon. It is peptide hormone. Peptide hormone which will be produced by pancreas. Very, very important. That increases glucose levels. Next. Which of the following statements are true about carbohydrates? So monosaccharides can be hydrolyzed. That is wrong statement. Two monosaccharide units obtained on hydrolysis of disaccharide can either be same or different. That is correct. Polysaccharides are no sweet in taste. That is also correct. All monosaccharides are not reducing sugars. That is incorrect. Therefore, 2, 3 are correct. Option B. All monosaccharides are reducing. All Monosaccharides are reducing sugars. Remember this point. Only 2, 3 are correct. Next. Identify the product of the following reaction. That means C6H10O5 N times, which is polysaccharide, undergoes hydrolysis 393 kelvins and 223 atmospheres. It gives n moles of C6H12O6 glucose. Therefore, option B. Simple question. So this is polysaccharide. Polysaccharide general formula C6H10O5 N times. Next. So which among the following acts as a vitamin? So ascorbic acid. Ascorbic acid is vitamin C. Vitamin C, therefore option B. Next, glucose does not react with which of the following? So glucose does not react with NaHSO3 due to absence of free aldehyde group. Due to absence of free CHO group, aldehyde group. Remaining reagents can react with glucose. So because of strong nature, strong reagents, those reagents can break cyclic structure of glucose. So NH2OH hydroxylamine can form oxime, HCN can form cyanohydrin, and bromine water can form gluconic acid. They can react with cyclic form of glucose. So these reagents can break cyclic structure of glucose, but NaHSO3 mild reagent. Therefore, it cannot react with glucose. Option D. Next. So general structural formula of alpha amino acid is given. The group R is in tryptophan and histidine. So direct the question. Option A. Option A. So here benzene ring, and here 5-member ring. Here CH2 and here NH. This is X. Next Y. 5-member ring. Here CH2, here NH, and here N, 2 bonds. This is Y. So option A. Next. So which of the following is structure of lactose? You know, lactose is so beta D glucose, beta D galactose, which is C1 attached to beta D glucose C4. Linkage between C1 and C4. C1 of galactose attaches to C4 of glucose. 1,4 linkage. which is glycosidic linkage. Option C. Next. So thyroxine produced in the thyroid gland is an iodinated, that means iodinated derivative of. Iodinated derivative of. So simply tyrosine. Iodinated derivative of tyrosine. Thyroxine. Direct question. Next question. Sucrose is dextrorotatory, but after hydrolysis the mixture shows levorotatory because of. So glucose, dextro, and fructose, levorotatory. So in this we write values. So here we write values. Glucose is dextrorotatory, which is plus rotation. So plus 52.5 degrees. Next, fructose is minus 92.4 degrees. So negative rotation is more than positive rotation. Therefore, inversion of cane sugar takes place, which is D. Levo rotation of fructose is more than dextro rotation of glucose. Next. So, which institute has approved emergency use of 2-deoxy-D-glucose as addictive therapy for COVID-19 patients? So, simply Drug Controller General of India. Drug Controller General of India. DCGI. Direct question. Next. So RNA, DNA are chiral molecules. Their chirality is due to the presence of D sugar. So D sugar means, wait. So here D sugar is ribose and deoxy or 2-deoxy ribose. So due to sugar is optically active, chiral. Option A. So ribose is present in RNA, deoxyribose is present in DNA. So due to sugar, it is chiral, optically active. Next. So C1-C4 glycosidic bond is not present in C1-C4. So simply sucrose. In sucrose, C1-C2 linkage is present. C1-C2. C1 of glucose and C2 of fructose. Very important. In remaining cases, C1-C4 is present. Next. The vitamin which helps in clotting of blood is vitamin K, simply. So deficiency of vitamin K causes coagulation. Slow downs the coagulation of blood. Nucleic acids. The nucleotides are joined together by phosphodiester linkage, clearly. Nucleotides are joined by phosphodiester linkage. Direct NCERT question. Option B. So nucleic acids are polymers of nucleotides, simply. Next. The two forms of D-glucopyranose are known as anomers. Anomers are alpha D glucopyranose and beta D glucopyranose. So these are differ in configuration at C1 carbon, only at C1 carbon. Therefore option B. Next, hormones are secreted by ductless glands of human body. Iodine containing hormone is iodine containing hormone. So clearly option, one second. So iodine containing hormone is thyroxine. Thyroxine. Iodine containing hormone. So option D, here it is wrong. Thyroxine. THYROXIN. Thyroxine is iodine containing hormone. So this is the end of the chapter. So in next video I will explain one more topic. All the best and thank you.